The next one is given by the Kim-Sara Wagner. So let me introduce Kim. Kim obtained his master's degree in biological oceanology from the University of Kiel. Now she is working as a doctoral researcher in the research group of parental investment and immune dynamics at GEOMA. And her research interested including evolutionary ecology and microbiology and host microbe interactions. In her studies, she mainly used molecular and microbiology approaches to explore the role of the microbe in pregnancy evolution. So a little thing that is a fun factor is that she is so addicted to Kung Fu that she sacrificed his living room to create an Asian-style training room. Oh, so that is very cool. So today she will present her ongoing PhD project about the sex-specific parental transfer of microbiota. So Kim, welcome. So go ahead, please.
Thank you very much. Let me quickly share my screen. Okay, can you see the screen or it? Sorry, just some small. Okay, again. Okay. So. Good afternoon, everyone, and thank you for this nice introduction. So as introduced before, my name is Kim and I'm a second-year PhD researcher in Olivia Rhodes' research group at GEOMA in Kiel, Germany. And today I would like to introduce you to the world of microbes and their role in male pregnancy evolution. When we hear of bacteria and microorganisms, we often think of pandemics, diseases, or catastrophes. For example, major diseases and pandemics like the cholera pandemic that struck Europe in the 19th century. And of course, the most recent pandemic and the reason why most of us have to attend this meeting virtually, unfortunately. But also minor health problems are connected to the occurrence of bacteria, for example, skin rashes or tooth decay. But in fact, microorganisms are more than just cruel pathogens. They are fascinating because they show an extreme diversity which allowed them to inhabit all habitats on Earth, ranging from extremely cold and hot environments or habitats that lack completely oxygen. And a large proportion of these microorganisms is actually non-pathogenic. Even more, they are beneficial for their host, with which they exist together as a unit, the so-called meta-organism. And this unit is shaped and influenced by the environment. And this is when we speak of the microbiota, an ecological community of microorganisms that includes all bacteria, viruses, fungi, archaea, and eukaryotes that inhabit an organism. So let's take a closer look at the microorganisms that each of us hosts on and inside their body. For a long time, it was believed that we have almost 10 times as many microbes than body cells. Of course, this number is hard to estimate because the number depends on sex, age, weight, and height. More recent studies suggest that we have almost equal number of bacterial cells than we have body cells. In vertebrates and invertebrates, a complex microbiome brings immunological, metabolic, and behavioral benefits, which in turn also means that disturbances can contribute to the development of disease states. The microbiota comprises much more than the frequently cited gut microbes. The microbiota also acts in the mouth or the skin, where it contributes to the protection, development, digestion, metabolism, and immunity of the host. However, the molecular and cellular mechanisms controlling these interactions within the meta-organisms are poorly understood. Also, many key interactions between the associated organisms still remain unknown. So for now, let's dive into one of these functional interactions, the immune system. The immune system exists in a constant interaction with the microbiota. Here, the microbiota produces vitamins or polysaccharides, which boost the onset and maturation of the immune system. The host immune system then regulates the growth and composition of the microbiota. This balance interaction is what we call a homeostasis and also describes a constant coevolution between the microbiota and the memory-based immune system. Here on the right side, it is nicely shown by Lloyd and Marsland how the immunity microbiota interaction changes in the course of life. This interaction is influenced by many external factors, for example, the exposure to pollution or the presence of pets, and also general life choices such as diet or smoking. Environmental factors, especially during early life, can induce long-reaching consequences for immune development. However, this relationship is very dynamic, and short and long-term fluctuations in microbiota can occur that affect the immune function. Knowing about the huge impact that microbes have on our bodies and lives, how do these microbes reach their host in the first place? Here we speak of primary colonizers. Primary colonizers are facultative anaerobic bacteria which colonize a host and then adjust the environmental conditions so that further colonization by other commensal microbes is made possible. In mammals, this primary colonization happens via the initial transfer of commensal bacteria during three major events, namely pregnancy, birth, and breastfeeding. And at this point, it is quite obvious that these processes are unique to the female body. So for a long time, it was assumed that the transfer of microbiota and eventually immunity occurs via the mother only. But nevertheless, the father should not be forgotten at this point. Let's talk about the paternal effect. The paternal effect is a non-genetic factor that is transmitted from a male to his offspring, which then has an effect on offspring development. The nature of this non-genetic factor is influenced by the paternal genotype, the environment, or both. And examples for such father-offspring influences are, for example, the ejaculate-borne defensive compound transfer in certain insects, the direct transfer of antimicrobial compounds to brooded eggs, or simply postnatal paternal care. In our working group, previous studies have shown that the maturation of the offspring immune system occurs through the transfer of immunity from both parents, father, mother, and also in combination, through the sex-specific transfer of immunological compounds. And due to the strong connection with the immune system, we now aim at showing the same biparental transfer, but in this case, for the microbiota. So why do we need signatures here? In classic female pregnancy system, the contribution of mother and father is very difficult to determine because egg production and pregnancy are entangled within the female body. In pipefish, however, we can disentangle the contribution of sex via the eggs and parental investment through its male pregnancy on offspring microbial composition. So this is the perfect model organism to study pregnancy evolution and to investigate sex-specific contributions within the transgenerational microbiota transfer. In my PhD research project, I focus on three aspects. I want to understand the role of the sex-specific microbiota in male pregnancy evolution. Also, I would like to look at the impact of the microbiota on offspring immune system, survival, and development. And I would like to find and highlight potential links to transgenerational immune priming, which is seen as a major component of parental investment. Because after all, my main goal is to investigate how male pregnancy has co-evolved with adaptive immune system rearrangements and the brood pouch specific microbiota. The first step to understanding the sex-specific microbiota is to check which microbes we find in the pipefish. A previous study of our research group, published in 2019, used culture-independent 16S rRNA sequencing to characterize the sex-specific microbiota composition in female pipefish, as well as non-pregnant pipefish and male pipefish during early, mid, and late pregnancy. Here on the left side, you can see a heat map of the most abundant OTUs, which is used to identify the connection between occurrence of most abundant species and the surface of the gonads, fertilized eggs, or embryos of increasing male pregnancy. On the right side, you can see a biplot which is an underlying principal component analysis of bacterial beta diversity to show bacterial community structure, which is then overlaid by a factor map, the arrows here, to show which bacterial species contribute the most to the observed pattern. In my master's thesis, I built upon this previous study and cultivated the sex-specific microbiota, which I then characterized by 16S Sanger sequencing. Here on the left side, you can see the absolute numbers. And in terms of these absolute numbers, bacteria of the genus Vibrio were most often cultivated, followed by Shawonella and Pseudo-Ultramonas. Here in the middle, we look at relative abundances. I was able to show that certain bacteria are specific to a sex or pregnancy stage. So each color here stands for a tissue or pregnancy stage. And to give you an example, all bacteria genera, for example, this here, where the red bar, so the female X is missing, means that this genus was only found in male pipefish. I also performed a principal component analysis to show the bacterial community structure and created an overlying factor map showing which species contribute the most to this pattern. And you will now understand why this complex biplot was useful, because when we reduce both biplots, the culture independent and the culture dependent one, to displaying only the clusters of pregnancy stages, we can see that we have very similar patterns between these two sequencing approaches. On both sides, we can see that there is a difference in microbial composition between the eggs and the brood pouch. However, doing non-early and mid-pregnancy, there's not such a big difference in community structure, but then at the very end, there's a drastic shift in composition. And it was shown in both approaches, which is pretty cool, in my opinion. Additionally, we identified an overlap of bacterial genera, Epibacterium, Marinomonas, Pseudomonas, Regeria, or Kiloniella, that are transferred only by male pipefish and can therefore be seen as paternal indicator species. Now we know how the microbiota in the pipefish looks like. A way to understand its function is to manipulate it. But for manipulation, we first need a depletion of the natural pipefish microbiota. In this experiment, I applied two different broad-spectrum antibiotics, chlorphenicol and kanamycin, separate and in combination to pipefish in both high and low doses. Using sterile swabs, I quantified the diversity of the sex-specific microbiota over time, seen here in hours and start. First results show that antibiotics, especially chlorphenicol, the purple line here, are indeed able to deplete the microbiota. The red line here shows the time when we reduce the doses from high to low. However, the lowest bacterial diversity occurred much later, which then underlines again the importance of giving enough time for an antibiotic treatment to show an effect. Next, I wanted to take a closer look at the previously identified paternal indicator species, named here again on the right, on the left side. Each plot displays the relative abundance of one of the bacterial genera over time in the 4 different treatments, green showing control and the other 3 colors showing the antibiotic treatments. In almost all cases, the antibiotics, does not matter which one we take, successfully depleted the strain. Only for one strain, Pseudomonas here in the right corner, we could not find any reduction in abundance over time with an antibiotic treatment. With this knowledge gained through previous studies and experiments, so the sex-specific microbiota composition on the one hand, and mechanisms of natural microbiota depletion on the other hand, I'm now actively manipulating the transfer of the sex-specific microbiota from pipefish parents to their offspring. This allows us to study implications for the male pregnancy itself, but most interestingly, we can now investigate the impact on early microbial colonization, offspring development, also the survival of the offspring, and the development of the immune defense. Last but not least, I'm also working on different fluorescent labeling techniques to visualize microbiota transfer across pregnancy stages. Here, I will add labeled bacteria strains to either the environment or the fish itself before mating, and then hope to relocate these bacteria in later stages of male pregnancy. Illustrated here, for example, the late pregnancy stage, in which I then aim to find labeled bacteria that originate, for example, from the eggs or the non-pregnant male brood pouch. I hope I was able to give you a good overview of my ongoing research about microbiota and male pregnancy. Here, I would like to thank my working group for their input, assistance in field and lab. And I would like to thank all of you for listening.
Okay, thank you. Thank you, Kim. So anyone has questions? Oh, Professor Mayer have one question. Okay, I'll open your voice. Oh no. No, I have a small question, Kim.
Yeah.
So you have compared the microbiota of the pepfish in different stages, and I was wondering, since the pepfish you start the brood pouch of it is not fully enclosed, and the sea houses have fully enclosed the brood pouch. So this is just explanatory questions. So what can you tell me what is the difference between the sea houses and the pepfish's microbiota? Can we compare the two species together?
So just to clarify, so you want to ask how the microbiota differs between the seahorse brood pouch and the pipefish brood pouch?
Yeah. Have you
So of course, sorry?
Have you did this?
I haven't done this, but this is done by a postdoc in our lab who is comparing microbiota across different levels of parental investment. But this data is currently still in analysis. So I'm only focusing on the different levels, the different stages of pregnancy in the pipefish.
Okay, thank you. Any questions? Please raise your hand, please.
So actually, you can now unmute yourself if you want to have a question.
Okay.
Thank you, Kim.
Okay.
Sorry.
Okay.